Hello friends, it's me Dhanashree again from Sri Science. Today, before getting into our lesson, I would like to ask you two questions, and they are: what is the organ present in our body that doesn't grow and remain in the same size till we die? And my second question is, name two organs that grow till our death. If you know the answer, please give your answers in the comment box below. So let's get back into our lesson. In this test also we have an aim, and that aim is to show that sucrose is present in the sample solution taken for the test. And here also we need some equipments to do the experiment, and those equipments are boiling tube, Benedict's reagent, sample sugar solution, Bunsen burner, throw with water, tripod and gauze, and top hydrochloric acid, and the base substance sodium bicarbonate. Okay, let's get into the experiment. Okay, I said this experiment is for the one which is a non-reducing sugar. So if we do the test that we did for reducing sugar, there should be no observation. So first let's do the test what we did for the reducing sugar. So take a boiling tube, add 2 cm cube of sample solution or a volume you wish. Then add Benedict's reagent of 2 cm volume or same volume as the taken sample solution, and then mix it very well. And you know the next step. Heat the solution at 95 degrees Celsius or boil it. Now, what will be your observation? Yes, there will be no color change, and you know the reason why. Because the test we did now is for reducing sugars, and as sucrose is a non-reducing sugar, this test won't answer for sucrose. So this proves that sucrose is not a reducing sugar, so it's a non-reducing sugar. So let's do this test again, but let's make some changes. Again, same procedure. Take a boiling tube, add 2 cm cube or a volume you wish of sample solution into it, and then starts the different procedures. You have to add hydrochloric acid, which is an acidic substance of volume 1 mm into the sample solution. Then you have to boil this for a few minutes. After you boiled it and after it cooled down, you have to add the basic substance sodium bicarbonate, because when you added dilute hydrochloric acid, this solution became acidic. So to neutralize it, you're adding a basic substance sodium bicarbonate. Now, no problem. Again, add 2 cm cube of Benedict's reagent and then mix it very well. And now, you know what the next step will be. Yes, take the boiling tube which contain the sample solution with a mixture of hydrochloric acid, sodium bicarbonate and Benedict's reagent, and immerse it into a trough with water, which means water bath. Then heat it at 95 degrees Celsius or boil it. And now, what will be your observation? Yes, you will observe a color change. Blue to green to yellow, orange, red. Yes, the same color change that you observed in the test for reducing sugar. What really happened in this experiment? Because in your first procedure, you didn't get any observation. There was no color change. It was in the same blue color. But in your second procedure, you got a color change. Then what happened in this experiment? Yes, when you added hydrochloric acid, this hydrochloric acid broke our sucrose into glucose and fructose. Now, when you add Benedict's reagent and heat it, there will be a color change. You know why? Sucrose and fructose are reducing sugars and they are monosaccharides. That's why when sucrose was in sucrose format, we didn't get an observation because it's a non-reducing sugar. But when the sucrose broke into glucose and fructose, as these both are reducing sugars, when you did the experiment again, you got an observation. And know one thing, we didn't use concentrated hydrochloric acid. We used dilute hydrochloric acid. Why? There is a reason. Because concentrated hydrochloric acid will dehydrate the carbohydrate sucrose. Yes, sucrose is a carbohydrate. So it contains water molecules and carbon. When this concentrated hydrochloric is added, it will remove all the water by vaporizing them and carbon will be the balance. That's why we don't use concentrated hydrochloric acid but dilute hydrochloric acid. Note, in this experiment, we can use the enzyme invertase or sucrase instead of hydrochloric acid. What did this hydrochloric acid do in this experiment? Yes, it broke down our sucrose into glucose and fructose. These enzymes will also do the same thing. But if you add these enzymes, you shouldn't heat or boil the boiling tube because it will inactivate our enzymes. Therefore, you shouldn't boil the solution after adding this enzyme. But later, you can boil after adding Benedict's solution. Anyhow, we don't use this enzymes in our test in laboratory. Why is it so? There is a reason for this also. Because these enzymes comparatively take more time than hydrochloric acid to break down sucrose into glucose and fructose. For example, if hydrochloric acid takes 1 minute to break down sucrose, this enzyme takes 10 minutes. That's why we don't use these enzymes mostly for this test, but you can use it still. So I hope you understood our today's lesson. And if you have any doubts, please don't hesitate to contact us through these addresses. And it's me, Thana Uma again, leaving from you till we meet in our next video. And friends, don't forget to answer the questions which I asked you in the starting. And I will give you the answers in my next